All right, hi everyone. My name is Aaron Baldwin. I'm an associate director at Microvention in the R&D department. I want to start off first for thanking the organizers for giving me the opportunity to introduce the X Technology surface treatment to everyone. From here, I'll start discussing the FREDEX device. To begin, the FREDEX device is mechanically identical to the FRED. We took the FRED device and we covalently bonded a surface treatment to the surface of the FRED. The surface treatment itself is a solution-based process that treats the entire surface. So the inside and out of the device is treated. The surface treatment is roughly 10 nanometers thick, and the surface treatment does, compared to the smallest fiber wire, it's roughly 21,600 nanometers. So it's roughly 0.05% of the smallest fiber wire of the device. The delivery and retraction force is reduced by the surface treatment. So here we can see a plot comparing FREDEX to FRED. So the advancement is reduced by 31%, and the retraction force is reduced by 38%. So from here, I want to go into a little bit about the background of the X technology treatment. There's been about 30 years or more of continued use and studied, first by Terumo. So this began back in the 1990s. Terumo first investigated this polymer that we use, and they roughly did about 11 years worth of research and development before they first placed it on their first devices. So clinically, it was used in 2001. On any device that would interact with blood, so anything from pump heads to blood bags to tubing sets, anything that would touch blood would have the surface treatment on this. And they first coined the term X coating on the for the surface treatment. In 2014, they had their first semi-implantable device. It was an oncology injection port, so they surface modified the lead wire to increase the longevity of the device. In 2016, we began our own research at Microvention, modifying the X coating to be permanently covalently bound to the surface of implant devices. And in 2020, we got our CE mark approval for our FREDEX device. Looking at the X technology on the surface, it's very hard to visualize because the surface treatment is roughly 10 nanometers. So the only way to look at this is at a cross-sectional view of a filar wire, and we can only view it using transmission electron microscopy, or a TEM. So we take a cross-sectional view of the wire, and we have to sputter some layers on it to visualize the surface. So if you look into the very small yellow box, in that section, you can see the functionalization layer, and the functionalization layer sandwiched in between those sputtered layers that are not part of the stent, but they're just there for visualization on the TEM. You can see that it's 8 nanometers and 6.6 nanometers. So from here, I want to move on to a little bit about the actual mechanisms or characteristics of the polymer. So the X coating and X technology uses PMEA, it's poly-2-methoxyethyl acrylate. This is the homopolymer that is used on the X coating, and our X technology uses a copolymer of the PMEA, but it's roughly 95% PMEA, and we had to change it slightly just so we can covalently bind it to the surface. So what's interesting about this polymer is that the biological performance can be inferred from its hydration characteristics. And so beginning in the 90s, the fundamental research of this polymer, we started to learn some interesting hydration characteristics of the polymer. So just to talk a little bit more about the polymer itself, down below in the table, we have 3 polymers, and they have different equilibrium water content. So when they come into contact with water, they absorb a certain amount of water. So PMEA is our X technology. In the middle, it absorbs roughly 9 weight% water. And if you compare that to HEMA, which is a very hydrophilic polymer used in contact lenses, it absorbs 4 to 5 times more water. And if we compare that to a hydrophobic polymer, PEHA does not absorb much water at all. So the PMEA has this intermediate hydrophilicity. But what's really interesting is that this hydrophilicity is not the complete story of blood compatibility. Just because a polymer is hydrophilic doesn't necessarily mean it will be compatible with blood. So moving on, what happens to a polymer as it absorbs water? So the schematic on the right will show the polymer. When it first comes into contact with water or blood, it rapidly absorbs the water. That water then transitions into several different types of water. So the first water that gets absorbed is tightly bound. So it's held very tightly into the inside of the polymer. Its interaction with the polymer is very strong, such that it actually doesn't freeze. If you took this polymer, this hydrated polymer, and froze it, this water would never freeze. It's permanently liquid. Unfortunately, this type of water is not very good for biological reasons. Polymers or proteins in the blood, if it interacts with tightly bound water, it will denature. The second type of water that interacts with the polymer is what's called intermediate water. So this water, it absorbs to the water, it interacts with, it absorbs to the polymer, it interacts with the polymer, but it's more loosely bound. So it's transient, it's able to move around the polymer. And actually this intermediate water is what protects native proteins from interacting with the polymer or this tightly bound water that has been shown to denature proteins. And the third type of water that is absorbed into the polymer is considered free water. So this water does not interact very strongly with the polymer, and it can freely exchange with the surroundings. So what's important here is this intermediate water. This is what dictates blood compatibility. So having the correct amount of intermediate water will protect proteins from denaturation, and it will increase the longevity of devices that are implanted. So moving on with this concept, I want to talk a little bit more about this. So go back to our table here, we can see the equilibrium content of these three polymers. And if you further analyze this polymer, you can see that there's actually one polymer on here that has this intermediate water, and this is determined through DSC. So this was first discovered back in the 90s, and then further research discussed this more. So PMEA is the only polymer compared to HEMA and PEHA that has this balanced amount of intermediate water. So what does this actually mean? So first I want to go into talk about protein adsorption. So once a device or polymer comes into contact with blood, what happens to those proteins that are in the blood? And this is really dependent on the intermediate water concentration. So the research that was first done looked at fibrinogen. What happens to fibrinogen when it adsorbs to three different polymers? So if you look at PMEA, which is our X technology, HEMA, which is the hydrophilic polymer that's used in contact lenses, and then polypropylene is your typical hydrophobic polymer. So what was realized here was that the amount of protein adsorbed on the PMEA surface was lower than HEMA or polypropylene. So what this meant was that the total amount is lower and the protein adsorption is slower. So this means that the protein adsorption on the surface is controlled. So now that we know the protein is controlled, the adsorption is controlled, what happens to those proteins on the surface? And we want to look at the protein conformation, or its three-dimensional structure. So if we take, you can investigate this using spectroscopy that looks at what's the alpha helical content for that structure. So if you look at a native fibrinogen, it has about 27% alpha helical structure through CD spectroscopy. Now, if this protein interacts with our surface treatment, our X technology, there is high shape retention. So only 5% of that alpha helical structure is lost, which is very good. Now, if you compare this to those two other polymers that do not have a high intermediate water content, they have none, there is a high possibility that it's going to lose its shape. It denatures on the surface. So schematically, what does this mean? So if you have an unfavorable intermediate water content on the surface, proteins will denature, they'll adsorb to the surface, and they will no longer be recognized as a native protein, and platelets will adhere to the surface and become activated. So now that we know proteins will adsorb to the surface and then the conformation is important, the next step is what happens to the cellular adhesion? How does that impact the cellular adhesion? And it turns out intermediate water is very important to determining how cells will adhere to the surface. So a research paper in 2016 investigated the use of PMEA on the surface of materials to look at cellular growth and adhesion. So if you look at the table down here, this is confocal microscopy of several different materials. So on the far left, you have polyethylene terephthalate. This is your standard tissue culture plate. So cells, it's well known that cells will adhere and they'll grow on this surface. Now, if you compare this to taking that PET and surface treating it with our X technology or PMEA, it does not impact the cellular adhesion or growth on this material. Moving one more over, we have our HEMA. Surface treating that material with HEMA definitely impacts the adhesion and growth of the material of cells. The final column on the right is PMPC. PMPC is a phosphorylcholine derivative that is used in many different surface treatments that interact with blood. So if you look at this, it impacts the adhesion of cells and also the growth of cells. So if you look at the cellular counts at 1 hour, 1 day, and 3 days, the tissue culture plate, or PET, is very comparable to our X technology PMEA surface. HEMA does have some cellular adhesion, but the cells do not grow during this 3-day process. And if you look at the phosphorylcholine surface treated coupon, there is minimal cell adhesion and no cellular growth during this experiment. So from here, I want to sum up this intermediate water concept. So down here, we'll have a schematic. I want to point out cells and platelets and how they interact with the surface once fibrinogen and fibronectin is present, and how intermediate water impacts that. So the first diagram that comes up is our PET and another hydrophobic material, polystyrene. So these materials do not have any intermediate water. So when it interacts with fibrinogen or fibronectin, they both deform onto the surface, they adsorb readily, and two things happen. The good news is cells can adhere to this, so you do get cellular adhesion and growth, but the bad news is platelets become activated and they adhere to the surface. If you move on to the next extreme, which is our extremely hydrophilic polymers, and this is our phosphorylcholine derivative, PMPC, or our PEG, polyethylene glycol is another widely used surface modification. These materials have too much intermediate water. So when blood comes into contact with the surface, native proteins, fibrinogen and fibronectin, do not adsorb to the surface very readily. So the good news for this is that platelets are not activated, they do not adhere to the surface. But the bad news is that neither do cells, and this has been shown in the scientific literature. The final schematic is when you have this preferential amount of intermediate water. So you have enough intermediate water to prevent native fibrinogen from adsorbing or deforming onto the surface, so you do not get platelet activation. But you are able to get fibronectin to adsorb to the surface, so much so that you can get cellular adhesion, as has been shown. So if you want to look at how does this impact our performance of our FRED device, the first and the most obvious thing is how does it impact blood loop performance? So for this study, we took blood from healthy volunteers because we did take the blood out of patients. We had to heparinize the blood. We then load our stents into a tube and connect the tube together with blood, and then we circulate it in a device. So on the right-hand side of the screen, you can see the Chandler loop itself. So it consists of a 37-degree water bath, and the tubes are then loaded into the apparatus in the middle, and this is circulated. So after 1 hour, we take the stents out and we can look at what has adsorbed to the surface, what biological material there is. So below you can see 3 examples, typical results from this experiment. So on the far left is the FRED device. The FRED device does not have a surface treatment, and biological material readily adsorbs to the surface. This is a very dramatic change between the surface treated devices. So in the middle, we have our FREDEX device with our X technology. On the far right, you have a phosphorylcholine stent, which is the pipeline shield. So there's a large difference between surface treated devices and non-surface treated devices for the blood loop experiments. So we can further get into what happens to the blood as it circulates afterwards. So if we look at how many platelets are left in the blood itself. So in this plot, you can see the platelet counts. On the far left, we have our pre-control platelet levels. The control loop is after 1 hour. You can see the platelets do decrease slightly, but statistically not significant. Now, if you compare this to our surface-treated devices on the right-hand side, the FREDEX and the pipeline shield, it is indistinguishable from the control blood loop. However, if you look at the FRED device, the non-surface treated device, we have a reduction in platelets. So the platelets are becoming activated and they are adhering to the surface. If you move on to look at the blood work chemistry, we can look at the TAC complex, the thrombin-antithrombin complex. The pre-control levels are very low. After it's been circulated in an empty loop for an hour, we do get a little bit of that biomarker. And if you compare the surface-treated FREDEX and phosphorylcholine devices, you can see that it is not statistically different from the control, whereas FRED has a heightened level of the TAC complex. The final biomarker that we can look at is beta-TG. So as in the previous slide, the pre-control levels are very low. After the blood has circulated for an hour, the control level of beta-TG does increase slightly. And then compared to the surface-treated devices, FREDEX and Pipeline Shield, they're not statistically different from the control blood loop. And then our FRED, it has activated platelets, the coagulation cascade has begun, and the beta-TG level is higher. So now that we know our blood loop performance is better with our X technology, we want to move on to what happens onto the surface if we try to seed cells. So for this experiment, we basically repeated the experiments that were done in literature using our covalently bound X technology. So we look at coupons of nitinol, X technology functionalized nitinol, and then a coupon that has the phosphorylcholine derivative PNPC. So this confocal image shows that the nitinol itself has really good cellular adhesion and growth. The cells are spreading on the surface. They're very happy. When you take that nitinol coupon and surface treat it with the X technology, there is no difference between that coupon. And then finally, if you take the PNPC surface treated coupon, we have the same issues as what has been seen published in literature, where the phosphorylcholine inhibits cellular adhesion and growth. So just to summarize all these concepts that we have here with the intermediate water, the first step is that the X technology has this preferential amount of intermediate water, which controls protein deposition, preserves protein shape, and this is also what provides our lubricity. Moving on from here, this intermediate water concentration that we have sustains its hemocompatibility. So adsorbed proteins maintain their shape, and this minimizes platelet adherence and minimizes the blood coagulation cascade. And then finally, our preferential amount of intermediate water allows for controlled protein adsorption, which enables cellular adhesion and growth, such that we do not have any difference between our non-surface treated and surface treated devices. So with that, that's the end of my presentation. I want to thank everyone for taking the time to listen to me. Thank you.